So a non-huntingtin lowering drug is about to be approved, assuming there's no hiccups. Prilenia is the drug we were just talking about as they finalize their phase 3 data. Are small molecules like Prilenia and Annexon, and I think there's some others now as well, the future until CRISPR comes in, Obi-Wan? I know
I
you like Star Wars, so I had to throw that in.
yeah, Obi-Wan kind of goes back to the ancient texts, the ancient traditions of the Jedi, and I guess that's what CRISPRing out DNA would be. I think, hopefully it's not controversial to say that we've always said we need to try everything that has a reasonable chance of success. And I don't care who gets across the line or how many horses get across the line. We just need to get across the line. And what's more, as soon as we have success with something, we need to double down on our efforts to approve other things. There's certainly been, I think, for the past sort of 10 or 15 years, the world of Huntington's drug development has been divided into Huntingtin lowering and everything else. And that is sensible, I think, because Huntingtin, the DNA, the RNA message, and the protein, those are the three kind of first things that happen when the gene is causing harm. Everything else happens downstream, and the things that are downstream are generally easier to manipulate, but because there are lots of them going on at the same time, each manipulation in the downstream stuff is likely to have a smaller effect. The Huntingtin lowering is harder to achieve, but if you can achieve it, you've got reason to believe that should be beneficial for every aspect of Huntington's disease. So it's high risk, high reward, lower risk, potentially lower reward, but we don't know. And so far, nothing has worked. And we don't know what's going to come out of the Prilenia trial. It's a drug that hasn't had success in previous trials, but there's hope that by testing it at a different dose in different population, it might give us a different result this time. But I think it's a sign of the fact that we need to test all options that have a rational and safe basis. When we say small molecules, that's usually distinct. What that usually means is it's something that can be taken as a pill. So it's small enough to be absorbed from the stomach and then cross into the blood-brain barrier and influence the brain. Big molecules includes things like ASOs, the tominersen, the Roche drug, and the Wave drug are those ASOs as well. And those are gene silencing drugs. They can't be absorbed into via the bloodstream, so they have to be given by injection into the spinal fluid. And obviously that comes with its own risks. What's happened that I'm particularly excited about since we started testing these Huntingtin lowering drugs is that we now have small molecule Huntingtin lowering drugs. So it's potentially best of both worlds scenario. In other words, it's a pill that potentially lowers Huntingtin. And branaplam was one which didn't pan out, but actually that was a drug that wasn't designed for Huntington's disease in the first place. PTC 518 is another drug that's a small molecule Huntingtin lowering drug that's being tested. CRISPR is a whole other ball game because CRISPR basically involves introducing super complicated molecular machinery into the brain in the form of genetic instructions, which would have to be packaged into a virus, and the virus is injected into the brain, and then the brain gets infected with this set of instructions for making a machine that snips the DNA. And it's one of those things that's extremely easy to imagine being a perfect cure for Huntington's disease. All you need is a CRISPR machine that goes into every neuron in your brain, finds a big CAG repeat, snips out the bit of it that's too big and joins the ends together so that you don't have a Huntington's disease expansion anymore. And if that did exactly that and nothing else, it would definitely be a perfect cure for Huntington's disease. But the trouble is, we don't have CRISPR machines that are accurate enough to do that. And even if we did, we don't have a way of packaging them and delivering them to human neurons because the brain is very difficult to spread drugs like that through. Having said all that, in the year 2000, we would have said that the idea of injecting a strand of synthetic DNA into someone's spinal fluid to switch off production of the Huntingtin gene would be too difficult to ever accomplish. And actually, we did that in 2017. We have yet to turn that into successful treatment, but like what they said about Everest, it was impossible until someone did it. To answer your question, Josh, we should try all things that are possible, and we should also try very hard to make impossible things possible. And then we should try and do those as well. But we do have to prioritize. So I think whenever we're thinking about what trials we should be running, we need to think about the mechanism and how relevant it is to Huntington's disease. And then we need to think about the drug and how likely the drug is to achieve that mechanism. And then we need to think about what could go wrong. And that's what's done along the way with all of these drugs and treatments. But I think that's probably why so many of our trials, even if we're super excited about them, so many of them end up giving us the result we didn't want. Because actually, it's not that we're completely surprised, it's that we run trials because we've got reason to believe that something could work. We've got reason to expect that it will work, but there are always things that can go wrong. And unfortunately, everything will go wrong until everything goes right. And for a drug to fail, only one thing has to go wrong, but for it to succeed, like 100 things have to go right. But science is cumulative, as I've been saying since I was an embryo. As long as we learn from negative trials, we will always go to bed knowing more than we knew when we woke up, even if we have fewer brain cells.
That's great. Thank you. I think my next question actually feeds into that, which is what are the impacts? What impacts are we seeing, if any, on clinical trials from the new staging system?
Yeah.
And will this help achieve endpoints regulatory agencies are currently vague about for trial analysis?
So the new staging system is called the ISS, which is the integrated staging system for Huntington's disease. And in a way, it doesn't do anything, because all it is a different way of describing everyone in the world with Huntington's disease. And we actually already had a way of describing them. So it's a little bit like getting a pack of 52 cards and saying, these are the red ones and these are the black ones. But another way of saying that is, well, these are the aces and these are the twos and these are the threes. And those are different systems for dividing them up, but you've still got the same 52 cards. And the question is, what's the system that's most useful? And the traditional system that we have comes from back when you had to make a clinical diagnosis of Huntington's disease and there was no genetic test. But if you had something that looked like this was in like the 1970s, if you had something that looked like HD and there was no genetic test, but if it ran in the family in the right way, then that was Huntington's disease. And then the people who didn't have it in that family were at risk, and then they had HD, and then they had HD. And then when people started to study the disease a bit better, and once we had the genetic test, we could say, well, actually, we only need to worry about the people with a positive genetic test, and we divide them into pre-symptomatic and symptomatic. Actually, we use the word manifest, which means you've developed signs of HD, because the idea for research and for clinical care was no one had HD until a neurologist said, you look like you've got Huntington's disease. So from today, you have Huntington's disease. And I think hopefully everyone listening and watching will know that that's, to put it politely, extremely artificial. But it was very helpful and it served its purpose because it enabled us to start studying the disease and running trials. The reality is that Huntington's disease is, of course, a continuous process inside our brains and at the molecular level. And that protein is being produced in every cell in our bodies, not just from the moment of birth, but from the moment of conception. And so it makes sense that there is damage or there are changes that the gene is causing many years before onset. So the sort of clinical system is to say you're pre-manifest and then you're manifest. And then once you're manifest, you have a degree of impairment that comes from Huntington's disease. And that's the kind of UHDRS type staging system, which is stage 1, 2, 3, 4, 5, depending on basically what you can do. In other words, stage 1 is typically people who can work. Stage 2 is you can't fully work necessarily, but you can walk. And stage 3 is maybe you need a wheelchair, broadly speaking. There's a bit more to it than that, but that's the kind of traditional system. And then the next thing that came along was, what about these people who might have some very subtle movement problems, but they're not bad enough to be obviously HD, but they've had a positive test and their mood isn't right and they're more irritable and aggressive than they used to be, and something's not quite right. Surely we need to acknowledge that either is or might be Huntington's disease. And that's where this concept of prodromal Huntington's disease came from. So now we have pre-manifest, prodromal, manifest, and once you're manifest, you divide into 5 stages. And that's the current system. So why do we need a new system? The answer is that system is heavily focused towards conceptualizing Huntington's disease as what happens after motor onset, and also really only allows meaningful measurement of Huntington's disease in that manifest population. But what we actually know, if we want to develop drugs to treat and prevent Huntington's disease is it's probably better to go earlier, at least at the stage of testing drugs to show that we can achieve a meaningful impact, because an earlier brain is more likely to be savable or to have more neurons that can be saved, and also it may be more resilient to potential side effects of the drug. And the whole concept of a new staging system comes from discussions with the regulators like the FDA on basically what way of describing Huntington's disease would they be willing to think about when it comes to trials to prevent onset of Huntington's disease, but that would also be fit for purpose in testing drugs in people who have what we traditionally call manifest HD. And what it comes down to is basically it's borrowed from cancer and is staged from 0 to 3 on the basis of the presence of the HD expansion puts you at stage 0 at birth. In other words, the gene is causing mischief. It's probably doing stuff we don't want it to do, but it hasn't had any measurable adverse effects yet. Stage 1 is something measurable has happened. There's a biomarker change that can be used to determine that this gene is starting to do something we don't want it to do. And at the moment, the only biomarker that's got enough data behind it is brain volume measurement using an MRI scan. Stage 2 is you have some evidence that the disease is starting to affect the functioning of the brain, but that is not yet producing significant impairment or disability. So in other words, that's things like your performance on cognitive thinking tests, but not in terms of loss of ability to work or do household or everyday tasks. And then stage 3 is closer to the kind of current system of manifest HD, where the disease is now much more likely to be visible from the outside. And the good thing about that is that the FDA really likes the idea of these discrete stages, and also the transition from one stage to another is extremely numerical. Okay, it can be calculated based on things we know about you, like your CAG and your brain volume measurements and your performance on cognitive tasks. And it also is a system that can be adapted if better biomarkers come along. So if neurofilament turns out to be as good as we think it might be, then you could change the stage 1 transition criterion to be more driven by neurofilament as well as brain volume changing. So what does that mean for the design of trials? Essentially, when we think about trials, we've always wanted to do trials in as early as possible, but we also need to balance that with the need to detect potential benefit or harm of the drug. So if someone who has no signs or symptoms of HD takes a drug and then 2 years later they're still fine, it's impossible to know whether that was because they were going to be fine anyway, or they're fine because the drug worked. And so with things like nearly all of the drugs we've tested that we thought might slow the progression of HD, they tend to get tested in what we used to call early manifest Huntington's disease, but which we now call early stage 3 ISS stage 3 Huntington's disease. But if you think about something like the PTC trial, that trial was designed and launched before the ISS was published, but it was designed in conversation with the people who were designing the ISS. And basically the rationale that informed the ISS was also used to help design the criteria for the PTC trial. And notably, that trial is the first Huntington lowering trial that's actually deliberately enrolled people who didn't have an official motor diagnosis of Huntington's disease. So these are prodromal HD or what we now call ISS stage 2 Huntington's disease. And that's a big change. And the FDA is okay with it. They've indicated that if trials are conducted in this framework in the future, is much more likely to result in drugs being licensed that would otherwise, under the old staging system, not get licensed because the criteria for defining what constitutes late pre-manifest HD and an effect of a drug had not been agreed on yet. In some ways, it changes nothing, but in other ways, it changes everything, because it alters not only how we think about designing trials, but also it really encourages us to reflect on, at all times, on the actual biology of the disease, rather than the sort of convenient shortcuts that have informed the way that we talk about pre-manifest and manifest HD. And if it sounds depressing, the idea that you've got Huntington's disease from the moment you're conceived or the moment of birth, my sort of philosophical way to think about that is it's a bit like a credit card bill. it's inside the envelope whether we open it or not. But the key to solving a problem is to open the envelope and deal with the problem. And if this is a system acknowledging that there are effects of the mutation on the brain, if that's a system that helps us get preventative drug trials launched, and along the way gets more people involved in research, and therefore things happen more quickly, then ultimately that's the right thing to do.